Safety Assessment of Genetically Modified Organisms or GMOs. This video will be covered on the first one is the purpose of safety assessment of GMO. The second one is the principles of the safety assessment of GMOs. And the third one is the methodology of safety assessment of GMO products, which will focus on the substantial equivalence. And the final one is the example of the safety assessment of GMO products, which is the flavor saver. The main purpose of the GMO product safety assessment is to identify the new or altered hazards associated with the product as a result of the genetic modifications. If a new or altered hazard, nutritional or other safety concern is identified, further investigation is undertaken to determine its relevance to the human health. The principles of safety assessment of GMO products derive from the recombinant DNA organisms. Information required vary according to the source of the GMO products, which are plants, animals, or microorganisms. And the safety assessment of GMO products derived from the recombinant DNA organisms is conducted in three phases as follows. For the first phase, assess is conducted according to the information of the recombinant DNA organisms. When the results of the phase 1 assessment indicate the presence of possible toxins or allergens in the GMO product derived from the recombinant DNA organisms, it is necessary to carry out phase 2 assessments. Phase 2 assessment is conducted according to the following information. The first one is the safety assessment of possible toxicity, and the second one is the safety assessment of possible allergenicities. When phase 1 and phase 2 assessment of GMOs products derived from the recombinant gene organisms indicate that the available data are insufficient for thorough safety assessments, properly designed animal studies are requested on the whole products. One of the primary requirements in commercializing a genetically modified organism or GMO is the proof of its substantial equivalence with its non-GMO counterpart. In other words, substantial equivalence means that a new product such as a GM crop must be the same as the non-GM crop except for the traits that were enhanced, added, or removed through the genetic engineering. Substantial equivalence that has three scenarios what are envisioned in modified food or plants, such as the safety of consumption, safety testing which focus on the trait or case-by-case assessments, and the parameters of substantial equivalence is concerned about the first one, molecular characterizations, the second one, the phenotypic characteristics, next one is the key nutrients, and then it is the toxicants, and the last one is allergens.
The example plant in GMO is flavor saver tomatoes. That is about the reducing of the polygalactoronase gene, which gene is concerned about the form of red food. For the mechanism of this gene is about the breakdown of pectin. When flavor saver tomatoes compared with the non-GMO tomatoes, that show the long time to form more than non-GMO tomatoes. But the other nutrients in tomatoes are similar. For the nutrients in tomatoes is vitamin A, vitamin C, and tomatin. For the tomatin is the main nutrient found in the tomatoes, and this nutrient is non-different from the parental tomatoes. When detecting on the HPLC analysis, these are involved in key nutrients, which is one in the parameter of the substantial equivalence.